First, I'd like to thank Leslie for inviting me in the Simons Foundation. It's quite an honor to come here and speak. First, I'd like to thank my group because I'm going to be showing a lot of stuff that's a lot of their work is throughout this presentation. And really, the point I want to get across is a couple things. One is that imaging techniques now have evolved so much these days that imaging is no longer just recording. It's really the start of exploration. And the other part is that so many fields now are involved that you really do need, your portfolio of people that needs to work on imaging is vast. And I think that's what makes it really exciting. So I'd like to start with a little bit of a history of imaging to put this in some context. So in the early days, and that's some really early imaging, it was essentially sort of recording and drawing. And this showed up in numerous places in the famous Dunhuang Cave star map. And in fact, also, there was an astronomical event, which is a supernova that was seen in 1054 in this constellation. And this was Anasazi. So as equipment got better, so here's Galileo's telescope. Here's the Pleiades, which is also in Taurus. Still drawing. And here's a little better instrument. And this is one of the first real drawings of what was called the Crab Nebula. And as we move to modern experiments, we have better equipment. And here's, now it's photographic plates. But here's what a modern picture of that thing looks like. And it's pretty extraordinary because it's actually digital data from multiple different sources. And it really now allows people to develop and test theories in a much more comprehensive way. Now, I'm going to be talking today more about the inner universe, of which there's some early experiments. This is an amazing picture from the Cave of Hands, which is just stencil. And one of the early imaging pioneers was Da Vinci, so hand drawings. And this is probably the first medical X-ray, which is an X-ray of Wilhelm Röntgen's wife's hand. And this is what a modern CT image looks like, which is now digital data and 3D digital data. Now, my own field is MRI, so I'm going to be talking mainly about MRI. And this is actually the first MR image, which is amazing, actually. 2 test tubes of water. But here's what a hand looks like. And MR is really interesting. So you can build better devices, and here's better devices. This is something we built. This is a picture of a finger, and you can actually see, I don't have a pointer, but you can see tendons. This person actually had climber's hands, so damage to the tendon. And so as the tools have developed, so has our ability to collect a lot more data. So early imaging, here's some more modern imaging. So one of the key things here about MRI that I want to stress, and we'll get into more detail, is that MR, unlike CT, is sensitive to a really wide range of parameters. So for example, you can image flow. So this is a blood flow image in a hand. So the point is that from this, you have multiple devices, multiple ways to collect data. What perhaps not everyone realizes is that it's a multimodality, but it's actually the same scanner gives you all of these. So MR has the power to collect a wide range. You often hear people talk about, put them in an fMRI scanner. And I always think, well, no, it's actually an MRI scanner that collects fMRI data, which gives you amazing power to look at, for example, one person's brain in multiple different ways. Okay, so the idea is that you have all of this data now, and it's really just the beginning of exploring, and I mean exploring in whatever field that you happen to be in. So I'm going to talk mainly about what I think of, I call the 3 pillars of neuroimaging, and I'll start with anatomical imaging. And I would like to follow a storyline here throughout the talk, which is how I got into some of the things I got into. And one of my key points in this talk is you absolutely never know where science is going to lead you. And I think that's the most exciting thing. So I went to Shark Week, near Birch Aquarium. And this is not that long ago. This was the way they were doing comparative anatomy, which seemed to me primitive. So we just said, let's put a shark in and look at it. So we can slice it ourselves. Those colors, we call that segmentation. So we look at the different organs. and we can do that kind of stuff. But scientifically, what they're interested in is what is the morphology of the lambdid sharks, which are these large sharks, great whites, salmon sharks, and how does the muscle relate to its ability to swim, et cetera, et cetera. So these are biological questions that we can quantitate fairly straightforwardly. And this is what we call morphology. And this was done in the lab of the late and great Jeff Graham, who was actually a huge influence and forward-thinking guy. So the question then came, well, how do you normally do evolutionary biology? And maybe there's a better way, instead of doing traditional dissection, maybe do digital dissection. And I had essentially zero experience in what people were normally doing. And it really is important with something like this, where you have an N of 1. That's the only one. So if you dissect it, you have an N of 0. And this is at the Marine Vertebrates Collection in San Diego. So they have a big, rare collection of fish that are preserved. So this came out of the idea was the Digital Fish Library project, which was, let's scan the fish, make the data available to people, and non-invasively, you're not destroying anything, and then have people look at it. Now, by look at it, that's kind of where my group is most interested. Actually, here's some local examples. I was hoping they might be here today, but this is from the American Museum of Natural History, where they were looking at cichlids. So these are basic morphology questions that you can answer by looking at the internal organs. This is another one from the American Museum, which is looking at ponyfish, which is a little interesting fish that have light organs. And here you start to get a picture of where we might be headed, which is that you see a bunch of shapes, and visually your eye sees the different shape. But what we're going to be interested in is how do you actually quantitate those shapes? We're interested in quantitation, and what is quantitation? Now, just to show you sort of the power of some of this, this is actually something we did just recently. Some of you perhaps are familiar with, there's a living fossil, it's called, and it's a very, are people familiar with this fish? It's called a coelacanth. So this was 360 million years old, and it was rediscovered in 1938. And what's interesting about this fish is that it's sort of at the branch in evolution between where the land animals, where fish started walking on land. So it's called one of the lobed fin fishes, and very little is known about it. And there's pretty much unstudied. It does a really strange thing. It does a headstand when it feeds, and there's not normally light down there, by the way. And no one really understands anything about this behavior. So it turns out that we'd done an MRI of this. There was a specimen in a collection. And people had known that it has this kind of strange organ, and it's called the rostral organ, which is a pretty generic term. So we hypothesized that this kind of looks like an antenna. You just look at it, and it's right in the front of the head, and there's no other fish in the world that has this. So we've done morphology, we've done scanning. What we decided to do is model it as an antenna. And in fact, when you model it as an antenna, like a rabbit ears on the old TV, for those of you old enough to remember what rabbit ears are, it turns out it has a sensitivity spot right in front of its mouth. Now, what these things do is these are not fast fish. I think you could tell that, right? So it's upside down, and when it detects something in front, it opens its mouth and sucks something in. And the question is, how does it know when there's something in front? So this was the idea. So this is meant to show that already we're bringing in several different fields here of study, and imaging sort of allows that. Now, let's get back to the shark stuff. The Digital Fish Library project attracted a really phenomenal postdoc, Carrie Yopak, who was very interested in shark evolutionary biology. And sharks are unbelievable animals. They have 7 senses: hearing and taste, sight and touch. They have smell. This is the great hammerhead. The great hammerhead, that head, part of the function of that head is it's a giant antenna for olfaction. So this is actually something we did for National Geographic. That olfactory lobe is a foot long. We'll get back to that later. Lateral lines, when you see fish all swimming together, they sense pressure and they sense electroreception. So they're very, very sensitive to electricity. So in those pictures where you see, actually you can see the dots. I don't know, can you all see those little dots in the front? Those black dots are actually little tubes that go from the outside straight into the nervous system. So they're incredible animals. Now, they're much more evolved in a way than the coelacanth. And so if you look at the neural structure of a shark, it's very complicated. And shark biologists themselves, it's a really hard thing to start dissecting something and trying to figure out what's inside. So Kara's thesis was really fascinating, and this was in her PhD before she got to my lab, which was that shark brains all look different. And the reason they look different is that they evolve in response to their habitat and their lifestyle. So if you look at these just visually, they look really different, which is somewhat remarkable. This is sort of like a homunculus. And when she came to my lab, the idea was, well, they look different. Is there a way to quantify what we mean by different? And she was interested in particular in cerebellar foliation. So that's, you can see the foliation, we just mean the folding. Like the human brain is highly folded. Some of these that are lying on the ground aren't, okay? So what you have is different species that are really living very different lives, right? So some are lying on the ground, some, so incredibly complex 3D eating, motion, visual. They're doing a lot.
The great white's turn of speed is powered.
But oddly enough, something like the whale shark, and this is called a planktivorous pelagic. It's very slow. It's the largest fish in the world. And looking at its brain, we actually got one. It actually turns out to be highly folded, and it's not actually clear why that's the case. We actually had half a brain. Not something very different than human imaging. It's not that easy to get these samples at all. And so we're looking at brain organization and just see how they're laid out. That wouldn't be typically an interesting thing in human imaging, is how the brain's laid out. But one of the things is that this is really a difficult problem. When you start looking at species where you have one, or in this case, half, a lot of the techniques that sort of use templates for brains don't really necessarily play. All right, so the idea is, how do you look at neural structure in elasmobranchs? Elasmobranchs means sharks and rays, if I didn't say that. So we did imaging. And the idea is to look at these structures and quantitate it. So how does one quantitate brain shape? So one way that people typically do it is they'll take a surface and they'll fit a surface. Okay, that's fairly standard. That really just looks at a surface. And what we're really interested in, as you can see, there's an entire brain. So we developed a technique which actually you can think of, you can fit things with waves. You know, you can fit a shape with waves. And so if you do this, you can think of multiple cores being fit by waves. It's a very general way, numerically, to fit something. The important point of that is it gives you a number. And so the idea is then you can phenotype, and by phenotype it means sort of morphological characterization as opposed to genotyping, which is genetic. And here you can just fit something and it gives you, there's a certain mathematical entity which tells you how much you're going to fit it by. So here we're able to quantify what we mean by foliation level. So now when we talk about shark foliation levels, we have a number that we can put. Now the interesting thing is, and this was a really nice algorithm done by Dr. Galinsky in the lab, a very rapid algorithm. And here we have one brain where we just show it fitting it more and more closely, just to sort of mimic different brain structures. here's a hugely important message, though. So we started by looking at fish and didn't really realize where we were going to end up. But where we ended up was we came up with a very interesting clinical solution. So here's the brains of collaborator Susan Taper, substance abuse in teenagers. And one of the things you want to do is you want to look at the cortical thickness. And the cortical thickness tells you something about what's done with this substance abuse. Now, another point of this is that the ability to quantify foliations is actually something that they didn't really have in a good way. So it now actually gives you a new method. So we're starting to actually look at this data using this method. So it's a new way to quantify. So you never really know where things are going to lead. Sometimes they lead to interesting places. All right, now I'd like to talk a little bit about diffusion imaging. The first one is sort of morphology. This is going to lead us to connectivity, and then we'll talk about function. So I assume people are somewhat familiar with diffusion imaging here. So here, if you take ink and you put it on a piece of paper, the patterns look very different. And what that's telling you something about the microstructure. So it's reflecting in tissue paper, it's sort of jumbled. Whereas in newspaper, it's organized, okay? NMR or MRI is very sensitive to diffusion. And what that's actually reflecting is what's called restricted diffusion, which means the diffusion is being restricted by the underlying geometry, which means that if you can measure that restriction, you might be able to say something about the underlying geometry. So if you have a neuron or a neural tube here that has myelin sheath, et cetera, it's restricted. So things will diffuse along more than they will across. Okay? So here's the basic idea. Is that in gray matter, it's kind of like the tissue paper that's kind of all jumbled. And you're going to get something, we call this isotropic, meaning in every direction it looks the same. And in white matter, you'll get something which looks, that's just the 3D version of that shape there. And it's reflecting some underlying coherent structure, which is the white matter. And MR, what MR is able to do is just to measure this in different directions. And each of those arrows would be a whole different image. Okay? So MR can measure these. It just happens to be very sensitive to diffusion. And you can measure that. So here's the 3D version. I'm just going to show you what images like this look like. Each of those is an image of the same person with diffusion weighting along a different direction. Okay? So you just take all that data in every spot, you just reconstruct that little thing there. And the thing that it's diffusing along, that actually tells you something about the orientation of the fiber, which is interesting. And we also use the words so something that's spherical is isotropic. It's the same in every direction. Something that's, an ellipsoid is anisotropic. So here's a picture of a spot, and you can see where you create these little diffusion ellipsoids, and they point along the direction. This is what we call standard diffusion tensor. So it's actually kind of remarkable. If you take a measure of the anisotropy, and so it's conceptually very simple. If something is a sphere, it's isotropic, so the anisotropy is 0, and the more stretched out is more anisotropic. So this is just coloring it. The brighter white means more anisotropic. So this is actually kind of remarkable. You just take MR images that are diffusion weighted, you calculate the anisotropy, and you plot it, and it looks like white matter. It's pretty remarkable, actually. However, look at that spot there that's dark, and you know that there's a lot of white matter there. It's actually a really serious problem, and for those of you who are math and physics, this leads to some very interesting stuff, because the real problem is it's kind of a poor model. It's also the main reason why if you ask anyone in a hospital how much diffusion tensor imaging they're doing, the answer is 0, because it's pretty non-quantitative. That's not to say there aren't a lot of publications on it. It's just not that quantitative. Okay, let's get back to fiber orientation. What do you do with that? So here, an old drawing of the heart. We'll go through the centuries here. Here's an anatomical image of the heart, which doesn't tell you that much. But heart is an amazing organ, right? It's got these muscle fibers for contraction. If you take a diffusion tensor image of that, what you see here is, now on the right is anatomy, and there are sheets of fibers. Okay, this is not like neural tissues. This is sheets of muscle. And they change orientation as you go in the wall. So the colors are related to orientation, and there are these little things we call glyphs. And so you can actually see the wall structure, which is interesting. Now, what do you do with that? And here's a good example of how sort of diffusion tensor imaging and imaging in general sort of fits into the big picture. Here's our first whole human heart, excised. This is not done in vivo. A lot of information there. But it's just part of one picture of what's going on. So there's a group at UCSD, Andrew McCulloch's lab, which is doing patient-specific modeling of heart failure. And heart failure from cardiomyopathy, for example, if you have structural problems cause contraction problems, which are causing electrical problems, et cetera, et cetera. So there's a bunch of people working on a general picture of what happens in cardiac failure. And this is one tool to use it. But it's very, very useful because it gives you that fiber orientation. And just because it's MR, I want to show you what else you can do with it, just to emphasize the multimodality nature of it, is you can look at, this is a mouse model of cardiac failure. And you can look here, for example, at wall thickness and how that changes with a disease model. So they have disease models of mouse, and you can look at the wall thickness. What I haven't shown is you can actually measure things like shear, other things with MR. Okay, so fiber orientation now is interesting from a neuroscience standpoint and a disease standpoint because it's sort of the gateway to looking at connectivity. So what I've done here on the left is I've taken those ellipsoids and I've just made it into a line, so it sort of points along the major direction. And you can see, and your eye is a great integrator. So your eye is showing you that there's a path there, right? So what's a huge subject now is what's called tractography, is you connect up those lines and you get a track. Okay, so the good news is that's really interesting. You can reconstruct neural fibers non-invasively. That's pretty remarkable. Well, you also notice that those don't look right, okay? And part of the problem is that neural tissues are not that simple. They're not simple at all, in fact, right? So here's a rat white matter electron micrograph. And this is one of the so in MRI, one of the problems is that the resolution that you're looking at, typically, it's a good number to keep in your mind, is like 1 millimeter cubed. Diffusion imaging is probably 2 millimeters cubed. Well, neurons are like 10, 20, 30 microns, okay? So you're averaging over a lot of stuff, and you always have to keep that in mind. And this is a serious problem. So what happens is during that spot, you can think of all those crossing fibers as turning the newspaper into the tissue paper. You just have a big jumble, and everything looks isotropic, and that doesn't mean that there isn't stuff going on underneath. So what happens is you get these dark spots. So what happens when you have white matter damage? Well, in white matter damage, what happens is you ruin the white matter, and everything sort of becomes isotropic, right? Because it's no longer a fiber. So this unfortunate artifact of the processing mimics a clinical problem, and that's the danger. So remember I said that you can think of the diffusion shape as a tensor. So for those of you not familiar with tensors, it's very simply just the shape. So we can actually describe it with more complicated shapes, and that means just more complicated tensors. So here what we've done is you can describe that region. Now instead of having a fiber described by an ellipsoid, by something a little more complicated, described by a more complicated shape. So you can see those funny little shapes, and some of them sort of look like this. So that's going to be fibers that are crossing. This is sometimes called the fiber crossing problem for obvious reasons. And that allows you to track through this. So that's sort of where a lot of the field is. Now, why would you use this? Let's assume we can do this. Let's go back to this question from an evolutionary biology standpoint. And this is work that was done with Steve Kajura, who's really interested in olfaction and the wiring of the olfactory system. So simply put, if you have uh, an olfactory bulb that's a foot long, it probably doesn't make a lot of sense to have neurons that are going like this. It's inefficient. Everything else does. All vertebrates do. So the idea is the wiring makes a difference. So they did the experiment, actually, and this is, they actually showed, the punchline is that they showed that these are different than every other vertebrate. But it took 3 months to prepare this sample, and it's in 2 dimensions. And we're interested in 3 dimensions. But it's a very nice result. And this is done by Trish Meredith and Steve Kajura and A. Hansen, I don't know. So this was done at the National Magnet Lab. It's done at a very high field. For those of you who know magnets, a clinical magnet is 3 Tesla. This was 17 Tesla. And what we're able to do is, by looking at a very small piece of the olfactory organ, do fiber tracking to show that this is true, non-invasively. So I assume there's some people in here that are doing diffusion tensor imaging. Is that possible? Maybe. There's sort of a logical fallacy in diffusion tensor imaging. And that is that you estimate that ellipsoid, and it's sort of, everything is estimated the same everywhere. The basic assumption is that it's independent of everything around it. Then you do tractography, and the assumption is that it's related to everything around it. And those can't both be true. So one of the things we've been working on recently, and I'll just give a schematic diagram. This is San Diego. Is how do you incorporate local and global information? And for those of you here who are maybe mathematically oriented and interested in networks and pathways, et cetera, this is a problem for you to consider. So I can get to the ballpark anyway. The ballpark is where the baseball is. That's where I live. If I know ahead of time that there's some construction on some of these streets, then I don't have all the right pathways. I can't go the same. I don't have all the pathways that I can go. So I can take different pathways. The point of this diagram is that if I know something ahead of time about the structure of the problem, that prior information actually gives me a lot of information. And it has a huge influence on what I can say about the possible pathways. So we call this entropy spectrum pathways, and there's a reference there for it. The result of this, and this is some really great work done by Vitaly Galinsky in my lab, which is to be able to now incorporate local and global information, so to estimate the diffusion locally based on the tracks and vice versa. So it's all done together. And what's actually, this is mainly for the people who do, maybe in the audience who might be doing diffusion imaging. Those ink patterns I showed are called often the probability density function, or interpreted as a probability density function. And that's not what these are. These are what's called the transition probability. And what this technique allows you to do, actually, is here's an example where an arbitrary number of fibers going through any particular voxel can be reconstructed. And there are multiple lengths, so it's multiple scales. And this is not something that standard methods can do. Because, in fact, you can collect as much data as you want for a voxel and try and do something, and there's essentially a limit about what you can do. If you incorporate information about the surrounding architecture, you can do a lot more. But I'll just show you a cool picture. So this allows you to reconstruct really a much more complete picture of the entire brain's architecture. If you look at other techniques, there's actually a lot of gaps. There's a lot of places where the techniques fail. Because every time you give a talk on diffusion imaging, you'll have a physiologist come up to you and say, that's not what the brain doesn't look like 2 crossing fibers. There's a lot of stuff going through there. So this, I think, is an advance in the sense that it's trying to incorporate, it's a problem of inference where it's trying to incorporate local information. And so there's some of the results. And so on the left are anisotropy maps, and on the right are the tractography. And probably the most important part, if you look on the lower right, on the lower left, what it's doing is it's taking the main direction of fiber. This is what you would get out of a normal technique. But on the right, what's actually showing is, if you can look at each of these voxels, there's a spread in each voxel of orientations. And you can see that you're not missing anything. In fact, it's actually tracking through gray matter, which is very interesting, because gray matter is adjacent to white matter, and it informs how you do your estimate. And this is an autism study, so we're starting to use this with Dr. Axel Mueller, who's at San Diego State. And if you're going to use it for diseases, for example, where connectivity, and there's a lot, and autism is something that we like to focus on. There's two, really. There's autism, and the other real question is what happens in traumatic brain injury. In TBI, no one has any idea what's going on. I mean, you look at an image, an anatomical image, looks fine, and you know there should be some white matter disruption, and people can't typically see it. Okay, so the third pillar is functional MRI, and I assume people are somewhat familiar with functional MRI. Let's go back to torus. And this is actually, what I'm going to show you is not part of our work, but I just think it's such a great example of what fMRI can do. And really, it aims towards this idea of looking at brain networks. So looking at brain networks, again, if you're interested in math, this is a good time to be an MRI because people are starting to look at connections in the brain. So if you just stare at the picture on the right for a second, the brain is pretty amazing, right? It just turns that into a human for you. So what is the relationship between form and motion? So watch this video and then tell me what you think. Yeah, what do you think of that? What comes to mind when you see that? It's creepy, right? And this is interesting. If you read that, why is it so creepy? This is really interesting. So here's a robot. And this is work, by the way, that's being done by Aisha Pinar-Sagan at UCSD. It's also a robot. And that's a real person. So what's bothering you is that if there's a match between the form and the motion, it's okay. This looks like a human form, non-human motion. And this is human. And this is a really interesting concept called the Uncanny Valley, which is that the more something becomes human, the more comfortable you feel with it just until the end when it's just a bit of a mismatch. And you can probably guess from an evolutionary standpoint why that is. So what's interesting is what Aisha did is that there are really three systems here. There's the planning action, there's the visual perception and motion, and there's something that communicates between the two. And this result is just really interesting, which is that when you see the results from this, you see that there's a clear activation of that. So the point of this is that you have some really interesting ideas that you can test that tell us something about the way the brain is working its networks. And by the way, that's why they look like that now, because it's not nearly as creepy. Okay, so getting back to evolutionary biology, we like to complete the picture for the sharks. There's an old Tractography. And the last one is function. So these were experiments where the attempt was to do functional MRI in a living shark. I really don't recommend these. They're not easy experiments. Had to build a special device to image it and keep it comfortable and happy in the scanner. And here's some anatomical images. You can see the brains right there in the middle. And the idea was to, in a swell shark, to do a paradigm, which is to give it visual and electrical stimulus, very, very faint electrical. I mean, they have unbelievable sensitivity to electricity. And these are what's called task-based. When you're doing fMRI and you give something a task and you look for activity, that we call that task-based. Okay. We actually did see something in one of our specimens, and if you notice, that doesn't look like the thing that was. Doesn't look like the task. And if you actually look at the data, it's really interesting, which is that you see these spreading waves. And it actually looks like there's a wave source and there is a nerve there. So we actually don't know what to think about this. It's not unlike what people see in migraines. So I suppose one interpretation is the shark was having a migraine, but we don't really understand this yet. But the result is very interesting. So task-based, who I just mentioned, so what you do is what I'm showing here is just 4 points in a time series. The green square is that green square. It's one voxel. And the red lines are the task that was given. So like this, tap on, tap on, tap off. And you're basically looking for a signal that mimics the task. So the signal processing, as we would call it here, is actually fairly simple. Is there a signal that matches the stimulus? Are they correlated? So you can, if the shapes are the same, you can think of that as a 1. If they're completely dissimilar, it's 0. And mathematically, we call that a correlation coefficient. So it's pretty easy when you know what's going in to test. Well, now we're up to date now. So MR technology has really advanced, and MR scanners now can collect data incredibly fast. And what has been resurrected, which is an idea called resting state, where let's forget about the task and just see what the brain's doing when you're lying there. Now, we got interested because in the shark experiments, they're so hard to do. Why not just see what's happening while it's lying there? So we got interested in this idea of resting state. And this is a result from one of the many papers that's coming out now. So what happens is you lie in the scanner and you just see what the brain's doing, okay? And there are modes. I put those in quotes for a reason, because the processing here, these look very clean. It's actually an incredibly difficult problem. What does it mean to have a brain mode, and how do you analyze data where you don't know what to expect? And as you can imagine, as you're lying in the scanner, there's a lot going on, right? So we got interested in this problem. And for you network and math people, there's a huge amount of work here where people, once you have these activations, let's assume they're correct, how are different parts lighting up together? What are the networks of the brain? So there's a lot of people who are looking at networks in the brain. That's a big, big area. The way that it's processed is problematic. There's a lot of sort of ad hocery in the way that that's done, in part because it's just a very difficult problem. But going back to the problem that we used with DTI, where we thought, maybe we can incorporate something about the structure of the problem with prior information, which we called ESP. And that's a very general idea. It's looking at a grid and looking at relationships between points. And it really could be used for almost anything. In fact, here the grid is now not just space, x and y, but time. So our idea was, can you formulate a way to look at spatial temporal data? And so this is our attempt. This is new. This is some data from Tom Liu at the fMRI Center that had been previously been published. So it's very good resting state data. And it looks like, in fact, you can. So here's just the detection of modes in the brain. And we did 2 different people and just wanted to see if you could detect similar modes in both. And you can. And you get these very interesting spatial temporal patterns. What they mean, that's something we have to discuss with the neuroscience community, obviously, that we're working closely with. But these are very complex patterns that come out. But the interesting thing is you can quantify these modes. We have a meaning for what these modes mean mathematically. Okay, so what I'm going to end with is that you never know where science is going to take you. So what I'm going to show you is another type of problem that has space-time difficulties in interpretation that in fact uses the identical analysis. And this is video that I collected in, well, 2005. There was 2 tornadoes there. So the detection of tornadoes and tornado genesis is obviously scientifically very difficult, but it has huge social implications. Thunderstorms have a real anatomy. We can think of it as an anatomy, a real structure. The problem is people don't know which ones are going to form tornadoes or not. And they're obviously a very rapidly changing environment. But this is called really so tornado genesis is what triggers tornadoes is unknown. And I think you probably all know what the implications of that is, but here's Joplin, Missouri. So here is an example where you see what's called a hook echo, which on radar data is a signature of tornadoes. You often see hook echoes that never form tornadoes. These are taken by radars that are scattered throughout the state. But the social implications are obviously really important. So this is before, and that's after. So this was a really awful event. And there's a little bit of video of this, and it formed incredibly rapidly. And here's the warning problem. So the warning problem is, here's a bunch of radar data. Some form, some don't. And in fact, in certain states, one of the problems is that they get so many warnings that no one listens anymore. So this is another picture I took in Kansas, riding behind with my friend Josh Worman, who invented that Doppler on wheels. So what we did is we took some radar data and said, is there a way to look at the data in the same way? And this is another picture I took right before we drove away fast, which is the most studied tornado in history, which is the Goshen County, Wyoming. There were a lot of people taking measurements on this. So here's the results of that. And they are pretty interesting. People had hypothesized that there's something called the descending reflectivity core, which it seems our method detects. What happens is you've all seen that the famous example is when you have a skater and she pulls her arms in, and the angular momentum, it causes her to speed up. It's conservation of angular momentum. Speeds up. And one of the things that happens in these storms is that you get what's called vortex stretching. So if you take a vortex, and that's just a thing spinning, you pull it like this. This is supposed to be one of the mechanisms. So in fact, you can detect now the vorticity. Vorticity is just the rotation. And then we actually used the same diffusion tensor imaging tracking, except actually not even looking at a tensor, but just using the same methodology. So it's not really looking at flow lines in the standard way, but it's looking at parameter lines, so to speak, to form this interesting picture. And in fact, that allows us to dynamically look at what we think is a new way to look at the data. And this is, again, it's a quantitative picture because all of these parameters that we take, we can quantitate their time dependence, their volume, et cetera. So the important message is you never know where things are going to last. It started with Shark Week. Our dog loves Shark Week. And you really don't know where things are going to end up. And I think the message of imaging now is it takes people in multiple different fields and multiple different talents, physics, math, neuroscience, meteorology. And these problems will lead you to, I think, really interesting interesting ideas. And it really is, it's a brand new field really for exploration. So I think I'll end with that, and I'm happy to take questions.